earlier on in the, shall we say, the depressing part of the thesis, I was wondering, my goodness, this is so complex, no one will ever be able to make any progress. What I'm reading in 28 is that whilst you're focusing on a point in the life cycle of the fly, or this action has an immense, broad and systemic change in the fruit fly and the fruit fly population. Did I get that right? In other words, whilst it is very complex, you found something that is understandable and a point of intervention that has clarity to it, that has broad systemic effects.
Yes, everything you've said is true, but the real excitement is we can actually analyze these broad systemic effects. We can, because we are able to evolutionarily produce populations with different rates of aging and compare them to closely related control populations, we can in fact unpack all of that complexity. We can study it scientifically. Not only can we change patterns of aging, but we can use changed patterns of aging to analyze what's under the hood evolutionarily. What were the gene frequency changes? What were the changes in gene regulation? What were the changes in patterns of damage? What were the other features of physiological function that were changed in order to produce a longer-lived organism? All of those things can be studied transparently and effectively.
And is the fruit, I mean, people might say, well, Michael, it's a fruit fly, but I'm not a fruit fly. There are people who said I'm not an ape 100 years ago or so as well. Mm-hmm. Is there enough commonality for you to say, in looking at species that reproduce the way that fruit flies do, yes, there are broad rules that come out of what we now can see, and as a result of our investigation?
Great question, Rob. In 1984, I didn't have a good answer to that question, so I proposed that instead of using fruit flies to address these problems, we should use mice, which are much more similar to us. And thus I proposed that we apply this experimental evolution strategy to mouse populations. Well, about 15 years after that proposal about mice was published, circa the year 2000, we were able to completely sequence the genomes of both fruit flies first, and then second, humans. So now we can look directly from the genomics to see if there are in fact commonalities by reading off the DNA sequences of both species. And even more than just the sequence similarity, we can do genomic experiments in both fruit flies and humans to check whether or not in fact the controls work the same way. Now, the ways in which you study human genomics are, of course, completely different from how you study fly genomics. You don't, for example, make humans evolve in giant laboratories, contrary to some science fiction. Instead, what you do is you look at normal genomic-wide variation in humans and associate that with functional characteristics of people who have different patterns of age-associated disease, such as heart disease or indeed cancer. In that way, you can build these bridges very effectively and very powerfully using genomic tools. So for the 15 years after 1984, from 84 to 99, I was in fact very keen on applying the methods I pioneered with flies to mice. Since 2000, everybody pretty much knows that we can now go from fruit flies to humans and back again and check whether or not our inferences in flies are in fact valid inferences for humans, because we have this genetic, well, really genome-wide superhighway that connects research in fruit flies with research in humans.
Brilliant. Well, it must be a great boon because it does give you all this commonality. So the quick answer to my question is yes, what we know in fruit flies helps us understand what happens with similar species that reproduce in the same kind of way.
Yes, we can make those connections genomically, and in particular we can be certain, we can specifically exclude the cases where there isn't the commonality. We can detect the lack of